Welcome to this first video of 2 on amperometric transducers. And this picture by Galileo is maybe a little fitting, I don't know, kind of entertaining to think about. But Galileo was a prisoner, as they say, to thinking about astronomy in a certain way. And of course, we've discussed a lot and you've done some calculations related to potentiometric transducers, but amperometric are slightly different. And so we'll compare and contrast in the pages ahead. The 5 topics we'll touch on in the next few minutes are shown here. And basically what we're doing is they're dividing this video into 2 parts. The first part is going to take us up to sensor design characteristics, and then part 2 will focus more on the 3 electrode amperometric system. And then we'll talk a little bit about the sensor output using Cottrell's equation. We've already discussed that electrochemical measurements can be based on potentiometric, amperometric, or conductivity techniques. And this current lecture focuses on amperometric measurement. We find that the amperometric measurement technique gives us a broader array of measurands. We can take a look at blood gases, metabolites, DNA, and antigens and antibodies. And of course, this all ends up giving us clinical analytic parameters that can be used in patient care processes. Just to start with a comparison between the potentiometric and the amperometric, for potentiometric sensors, as a result of the specific sensor-analyte interactions, we got that local Nernstian equilibrium formed at the sensor interface, okay? And as a result, we talked about that last time, no current is allowed to flow in the system, giving information about the analyte's concentration. We're in a little bit different situation with amperometric sensors, and now we're actually doing is a voltage is placed between the reference and working loads to initiate an electrical chemical oxidation or reduction, okay? So now, instead of no current being allowed, we're measuring the resulting current, which is a quantitative indicator of the analyte's concentration. Again, the difference is in electrochemical methods, one measures voltage, other measures current flow. I would say that in the literature, sometimes amperometric sensors are sometimes referred to as a subclass of voltammetric sensors, but that distinction is not all that important to dive into right now. Let's spend a moment to compare and contrast amperometric to potentiometric. Of course, we just went through a lot of discussion earlier about the ion-selective electrode and the way in which these particular charge particles could be detected. We now have a slightly different situation, okay, where the transducer is a carbon electrode or chemically modified electrodes, and we get to things like measuring oxygen, sugars, and alcohols. And these are important primarily for glucose measurement. And of course, conductometric is a little bit different, and we're not going to really touch on that, but it's very powerful in urea and charged species as well. Just to kind of highlight this amperometric principle, spend a little bit of time on this possibly, but when the potential of the electrode-electrolyte interface is changed, and that is to say, with the appropriate completion of the circuit from equilibrium potential, all right, from equilibrium potential, the equilibrium of the interface is, of course, disturbed. What happens is in amperometric systems are based on monitoring the current associated with the oxidation or reduction of an electroactive species involved in the recognition process. So basically, any sensing scheme that imposes a non-equilibrium potential, remember we focused a lot about equilibrium of the interface with the potentiometric. Any sensing scheme that imposes a non-equilibrium potential and measures the current is basically an amperometric sensing scheme, and there's many like this. One of the fundamental principles in amperometric measurement is the mediated detection of current from oxidation or reduction reaction. Okay, it's a mediated process. So the principle of functioning is based on the application of a fixed potential to the electrochemical cell, resulting in a current because of the oxidation and reduction reaction. And again, as we've mentioned, this current is used to quantify the species involved in the reaction. So amperometric biosensors are often used on a large scale for analytes such as glucose, lactate, and salicylic acid. Very important, of course, in a lot of healthcare monitoring. You see that the enzyme in this case is a mediator, whether we're talking about the analyte in the top figure or in the bottom figure. 2 different sort of characterizations of it. But essentially, by changing the enzyme in the mediator, it is easy to extend the amperometric sensor to the analysis of other analytes. Let's take a look at glucose detection systems, which are really a classic amperometric example. The amperometric enzyme electrodes proposed by Clark and Lyons in 1962 was really a fundamental change, and that is based on this glucose oxidase, and it's played a leading role in the move to simple, easy-to-use blood sugar testing. Many people do their blood sugar testing at home, again, typically as part of their diabetes control. So not only, the glucose oxidase, but the other mediators have expanded amperometric applications as well. So you've got the, in this case, the glucose oxidase produces gluconic acid and hydrogen peroxide. So then subsequent reactions actually produce the electrons that would actually result in the flow that we're going to be measuring. So glucose remains one of the commercial success stories. The first generation of glucose biosensors used the glucose oxidase enzyme as a catalyst to study substrate depletion or production during the reaction. And again, depletion is a key word here because this means that there's going to be a life associated with the sensor itself. It's going to have a finite life. So the drawbacks associated with this type of system were really fluctuation in the normal level of oxygen and overlapping of background interference of the redox reaction with the hydrogen peroxide oxidation and the oxygen reduction. So a lot of development, of course, which was very important. And in the final generation of glucose sensors, the enzyme glucose oxidase was immobilized on a conducting polymer-coated electrode, and this facilitates the direct transfer electrons between the enzyme and the electrodes. So instead of being indirect, it is now a direct transfer of electrons between the enzyme and the electrodes. So the performance of these enzyme-based sensors relies heavily on the properties of the supporting materials and the immobilization techniques. So you can imagine there's a lot of different technology and ideas surrounding that. Much of that we're not going to go into. We're just sort of focused on the overall aspects of the glucose detection systems as an example of classic amperometric technology. Continuing on with glucose detection systems, there's various ways to measure glucose in aqueous solutions. In one form, an oxygen sensor that measures oxygen concentration that is the result of the glucose reaction, output current. So basically in this version 1, we really measure oxygen consumption, and it's a proxy for the glucose levels. A pH sensor that can measure gluconic acid and a reaction product of glucose would result a voltage change. So basically in this pH sensor, we're measuring acid production. And a peroxide sensor that measures hydrogen peroxide concentration that's also a result of the glucose reaction. And that there's a transducer that converts the concentration of the peroxide into an electrical current. So again, 3 different varieties here, but it's all about different techniques for measuring glucose sensors. And it comes down to really the fabrication, the cost, the accuracy, and the complexity that is of interest in the sensor design. Other amperometric applications rely on different substrates and bioreceptors, and of course the products that are detected. Ethanol, of course, we're going to have right here. The bioreceptor is alcohol oxidase, and the range is 3 grams per liter. So that's pretty accurate, pretty high resolution there. We just talked about glucose, and the bioreceptor is the glucose oxidase. And of course, look at the range here in terms of the accuracy. I think this has a lot to do with the actual fabrication and the nature of the sensor. So you can see that there's different ones. Lactate obviously has lactate oxidase as a bioreceptor, and the range is 40 grams per liter. So you get a variety of different concentration levels that you can detect based on the design of the sensor for given substrate, and of course the bioreceptor that's produced and the products that is detected. So sensor design characteristics, basically there's 8 parameters in the design. So as we hinted out just a little bit earlier, probably not too clearly, but basically the sensitivity is the response of the sensor to per unit charge in the analyte concentration. All right. Now the selectivity is the ability of the sensor to respond only to the target analyte. So basically, sensitivity, looking for responses to unit changes, analyte concentration, and selectivity is a response to the target analyte. Most people would know that. Biomedical engineers, of course, is a standard. But the range is the concentration over which the sensitivity of the sensor is good. Now, on our previous chart, we saw some range values. Some were narrow and some were broad. And of course, that specification would be part of the design process. Response time is the time required for the sensor to indicate 63% of its final response due to a step change in analyte concentration. So the time-dependent behavior and the way in which you analyze that, either digitally or analog is going to be important. Reproducibility, of course, is the accuracy with which the sensor's out can be retained. Detection limit, lowest concentration. And lifetime is the time period of which the sensor can be used without significant deterioration in performance characteristics. And why is that? Well, we mentioned it a couple slides ago. We talked about these as being reactions, and the way you've got the impregnated surface, the material that you're oxidation or reducing may diminish over time. And of course, last but not least, the stability characterizes the change in baseline or sensitivity over a fixed period of time. These 8 parameters are probably not news to many of you. Things like sensitivity, selectivity, range. I'm sure you've been through this before, but I just wanted to mention this as sort of a commentary on these specific sensors, just because it does matter in the design of the system. These are very thoughtful, and selection of parameters is intentional, not accidental. So the design consideration, we look at the process. It's kind of like this. Once the target analyte has been identified, the major tasks in developing the biosensor involve selection of a suitable bioreceptor or recognition module. This may not be easy based on the 8 criteria that we just outlined on the previous slide. Then after we get this suitable bioreceptor, now it's the immobilization method. Again, techniques vary, and that's going to have an impact on the sensor characteristics. Then last but not least, this design of the transducer that translate the binding reaction into a measurable signal. So really not too complicated here. You first off, you start by selecting a suitable bioreceptor. Then you move on to the way in which you're going to immobilize, and then last, looking at the selection design of the transducer to translate the reaction into a measurable signal. So the design of the biosensor, considering measurement range, linearity, and minimization of interference, and enhancement sensitivity, this is, I would have to say, having worked in this field for quite a while, often will tend to be trial and error. And once you're done with all that and you've got a system, packaging of the biosensor into a complete device. And what I don't mean here about packaging is putting it in a pretty box with a bow. We're talking about encapsulating the biosensor into a robust substrate so that it can be delivered and used by people. So again, just to kind of summarize, the first in this list requires knowledge in biochemistry and biology. Second is chemistry, electrochemistry and physics, and the fourth requires knowledge of kinetics and mass transfer. Once the biosensor has been designed, again, packaged for convenient manufacturing. We're not talking about printing up with a bow. All right, the knowledge of kinetics and mass transfer is going to be important because we're talking about the way in which molecules are whisked away from the surface of the substrate. We wrap up this portion of the video looking at the advantages and disadvantages of enzymes in biosensor architectures. Clearly, advantages are that the enzymes have very high catalytic activity, and this results in effective signal amplification. Of course, enzymes have a very high selectivity for their substrates. Very, very positive, clear advantage. Now, sometimes, as we've alluded to earlier, the improvement in enzyme stability is found when they're immobilized to transducer surface. So that decision in the design is going to be, of course, the function of the stability criteria and the need for it or absence. Disadvantages are that enzymes are fairly large molecules. And interestingly enough, you have this high catalytic turnover at the active site of the enzyme. But basically, the overall catalytic density is low. The volume produced is low. kind of interesting that you've got signal amplification, but the density is low. Often, the active site of the enzyme is deeply buried within the surrounding protein shell. Thus, the electron transfer is often not possible, and additional mediators required to affect the desired outcome. Enzymes also have a limited lifetime, so biosensors exhibit only a limited long-term stability, operational lifetimes of less than 90 days. This, of course, is fundamental to the reason for the shelf life of many of the glucose sensors that are offered in the market. So check the expiration date, right? Okay, so this concludes part 1 of the video lecture on amperometric electrodes. Our next video will pick up on the 3-electrode amperometric system and calculating sensor output with the Cottrell equation. As always, please reach out at any time with questions, concerns, or clarifications that you are requested.